Good morning, everyone, or good afternoon if you are in the same time zone as me. I think I need Belinda's help to reset my biological clock. It's not working very well these days, but I don't think I will ever try to adapt this time. It's going to be a short trip to the US, but I'm extremely grateful for the opportunity to present the work that we have been doing on the mechanisms of myotonic dystrophy in the brain. And a few years back, when I used to talk about the work done in the lab, I was worried to convince the audience that myotonic dystrophy was not just about muscles. I think today I don't need to twist your arm, thanks to the excellent work done by some researchers and clinicians, some of them here today, that myotonic dystrophy also involves the brain and is also a brain disease. But when you think about the brain, we usually think about neurons. And so to shake things up a bit and give myself another challenge, today my mission is to convince you that DM affects not only the neurons, but other cells that are important in the brain. So my talk will mainly focus on myotonic dystrophy type 1. Is this working? Yeah. Can you see it? Yeah. Myotonic dystrophy type 1, although I suspect that many of the findings that we have in myotonic dystrophy type 1 will be applicable to some extent to DM type 2. And I will review first some published data, and hopefully at the end I will have time to touch on some of the latest results we have in the lab. So as we heard yesterday, and in spite of its somehow misleading name, myotonic dystrophy is a multisystemic disease. It affects many tissues, virtually every tissue, every organ in the human body, with a profound impact on the central nervous system. That can lead to cognition, behavioral, and sleep defects. And this makes like a ripple effect that will make academic and professional success difficult, that may lead to socioeconomic deprivation, and then can have some effect and can cause some emotional struggles throughout life. Like all the other symptoms of the disease, the neurological manifestations come in a mixed collection, varying in severity and onset across generations, from a mild but very typical executive dysfunction in the adult form down to intellectual disability in the earlier onset forms of the disease. And this goes well in parallel with the phenomenon of anticipation that we heard yesterday from Darren during his talk. So I would say that I don't need to tell you these to review the RNA mechanisms in the disease, but after last night's show, I don't know what I'm supposed to believe anymore. And in case your mind was tricked by the musician, let me just go quickly through these very fast. So you know that the CTG repeat expansion leads to the accumulation of these abnormal RNA foci in the cell that interferes with RNA binding proteins, particularly through the sequestration and depletion of the MNL family. And this will end, but other disease intermediates have also been suggested to play a role in DM1 pathogenesis. And this leads primarily to the dysregulation of the alternative splicing of hundreds of transcripts in multiple genes and tissues. But also changes in polyadenylation and RNA trafficking within the cell have been suggested to play a role. And I don't think that I need to tell you that we know now that DM1 is a prototype of this toxic gain of function of RNA diseases, and that is increasing. And we've heard yesterday from the most recent cousin in the family, so the Fuchs dystrophy that has now been enlarging this family. And so what we learn from DM1 may have wider implications to the understanding of other human conditions. Although in spite of this progress and in spite of the fact that you know a lot about the fundamental aspects of the disease, we still don't know exactly how to establish the mechanistic link between the mutation itself and the onset of the neuropsychological manifestations of DM1. In particular, we do not know what are the cell types and even subpopulations that are most affected in the brain, and what are the molecular pathways that lead to the neurological symptoms. This is a critical question, of course, if you want to develop effective therapies, but it's a complex question. It is a complex question because the brain consists of many different cells that are highly heterogeneous and that have that can be affected by the disease in different ways and to different extents. Of course, we have the neurons that are the main communicating cells in the brain. They communicate through chemical and electrical signals. But we also have the astrocytes that have a critical role, not only functional, but also structural role, keeping the synapse in place, regulating neurotransmitters, ions, and providing metabolites to the trophic support of the neurons. We have the oligodendrocytes that are well known for the production of myelin, that is the insulator that wraps around the axon, speeding up the conduction of the electric signal. And we have the microglia that are our primary immune defense in the CNS that has been, and they have been involved in neuroinflammation in many neurological diseases. So DM1 may affect each one of these diseases, of these cell types individually, but it may also play a role and mess up the communication between them that is critical for proper brain function. So how can we study these mechanisms? One possibility is to perform RNA sequencing and transcriptomic analysis using human tissue. And this is one example coming from Eric Wang and his lab. They have done RNA sequencing in human frontal cortex, and they have found molecular evidence of defects in the trafficking of molecules within the brain cells, a molecular signature of neuroinflammation as well. And interestingly, most of the, if we concentrate on the genes that they found to be downregulated in the DM1 samples, there was an enrichment for genes that were somehow involved in the morphogenesis of the cells, in the cytoskeleton organization and dynamics. And this is interesting because this kind of goes together well with the cellular phenotypes that other labs have found, working either with the neuronal cell models derived from a transgenic mouse model. This is the case of Guichin Wang in Taiwan, or neuronal cells derived from human iPS cells or human pluripotent cells. This is the case of Cécile Martin's lab. They have found abnormalities in the morphology of neurons. So the cellular phenotypes go together with the molecular defects. But while the expression changes, in particular this downregulation, seems to concentrate on transcripts that are known to be specific on neurons, the splicing changes found in Eric's lab seem to have a more evenly distribution affecting transcripts that are expressed in different, or that are typically associated with different brain cells. Not only neurons, but also the astrocytes, oligodendrocytes, and microglia. So opening up the idea that this spliciopathy might actually be going on in different brain cell types. And we also know that the DMPK gene here, the human and the mouse, both of them, they are highly expressed in glial cells. Not only the astrocytes, but also different stages of the oligodendroglial lineage, from the OPCs to the pre-oligodendrocytes to the fully differentiated oligodendrocytes. So if they are mutant, somehow if these transcripts carry the CUG repeat expansion, they will more likely accumulate also in these other cell types and have downstream effects in the brain. So our idea was to use our transgenic mouse model that we have in the lab, and that I will tell you about briefly in the next slide, to look for the glial abnormalities, try to understand how they will impact the function of the neurons in the brain, and then to look for the mechanisms of RNA toxicity to explain these abnormalities. So the DM super cell mice that we have in the lab, and that most of you know about, express a large CTG repeat expansion that is inserted in the human transgene. The transgene is expressed in different tissues, including in the central nervous system and different areas of the central nervous system, which leads to the accumulation of toxic RNA foci in the nucleus of neurons, astrocytes, and oligodendrocytes, at least in the brain. And these foci co-localize with MNL1. I don't know if you can see it well, but they co-localize with MNL1 and MNL2. And homozygous mice, they produce enough toxic RNA to develop relevant phenotypes in terms of their behavior, but also in terms of the synaptic plasticity. And they also show some relevant changes in the levels of neurotransmitters that are clinically relevant maybe for DM1, in particular GABA. They have an increase in GABA levels in the brain. So they seem to offer a good tool to start dissecting this mechanism, what is going on in the brain, in the communication between brain cells, and in the mechanisms that are dysregulated somehow. So our first question was, does this toxic RNA affect the astrocyte biology in the brain of these mice? So first we looked at the astrocyte density in the brain to see if there were changes, and we could not see a different cellular density of these astrocytes in the brain of DM superXL mice. But another critical feature of the astrocytes is that they are extremely ramified in order to cover all the synapses around them. So to study their morphology and ramification in vivo, we have transduced them with a fluorescent protein and performed Scholl analysis. And if you look carefully here at the bottom, you see that the astrocyte of the DM superXL mouse is smaller, is less ramified than the control here on the top coming from a wild type glutamate, something that is quantified here in the Scholl analysis. And this phenotype seems to aggravate with age. If you compare what happens in the mouse at 1 month of age with 4 months, you see that the difference in ramification of the astrocytes seems to be more pronounced at later stages. And it is accompanied by a downregulation of GFAP, which is a cytoskeleton protein specific of astrocytes. Interestingly, though, this downregulation of GFAP seems to precede the abnormal morphology, this hypertrophy of the astrocytes, because you can see it already at 10 days, a time point when the morphology of the astrocytes is still unaltered in the DM superXL animals. We then moved to cell cultures to get a better insight about the morphology of the cells, and it will be easier to manipulate and to dig into the mechanisms behind these astrocyte pathology. And by live cell video microscopy, we could confirm the abnormalities in the morphology of the cells. So they were slightly smaller at all time points after plating. So they have this spreading defect. They don't spread as fast as the wild type cells. A phenotype that might be the consequence of the fact they assemble fewer focal adhesions, which are the contact points of the cell with the extracellular substrate and with the cytoskeleton inside. And the focal adhesions are, among other things, very important for the migration of the cells. And this is something you can easily assess in vitro. So to get further evidence of this astrocyte pathology, if you want, in cell culture, we performed a scratch assay. So you make a scratch on the monolayer cell, on the monolayer of cells, and you measure the time and you monitor the trajectory of the cells while they are filling up the gap left behind by the scratch. And when we did that, there was no overall difference in the speed of the front of migration. However, we noticed that the DM superXL astrocytes migrate more randomly. They are not as efficient in their migration as the wild type astrocytes. They migrate more randomly and more rapidly. They travel a wider distance during this displacement, but is not very directional. They are more erratic in their movements, possibly because they have weaker forces of attachment to the extracellular substrate. So I was quite interested by Christoph Stolk and the N-fix factor, and what we discussed briefly at the end about the possible role of N-fix on the migration of cells. So we'll have a look at that, Christoph. So why are they so affected, the astrocytes? Well, not surprisingly, also in culture, the transgene is more highly expressed in the astrocytes in all the time points that we analyzed so far in comparison to primary neurons. And they have more foci than neurons, as you could eventually see here. I'm not sure you can, but there are more foci in the astrocytes per individual nucleus than in the neurons. And these foci co-localize with MBNL1 and MBNL2, although I must say that in primary neurons and in primary astrocytes, MBNL1 and MBNL2 are primarily cytoplasmic. With all the antibodies that we tested, we find a pronounced cytoplasmic localization of MBNL1 and MBNL2. However, there is co-localization, and more surprisingly, though, there is a down-regulation of the total levels of MBNL1 and MBNL2. And we are quite sure about it. We thought it was our extraction protocol. We tried different extraction protocols, we tried different antibodies, and we see consistently a downregulation of the total levels of MBNL1 and MBNL2 in the primary DM superXL astrocytes in comparison to wild types. And this is something that also Rick Wansink has found in muscle cell models of the disease, and we don't know yet how to explain it, because the transcript is not the total levels of the transcript is not downregulated. In contrast, the total levels of self do not seem to change. We didn't assess phosphorylation, but the total levels of self remain unaltered in the DM superXL astrocytes, self1 and self2. So to gain insight into the role of these, the consequences of this MBNL sequestration and depletion in the astrocytes, we have knocked them out in wild-type astrocytes, and we could recreate the adhesion phenotype of the cells, here illustrated by the lower number of focal adhesions. So the MBNL seems to play a critical role in these abnormalities of the astrocytes that we are seeing. But then we wanted to know the targets, the intermediates and the mechanisms behind these cellular phenotypes. So we turned into RNA sequencing in our primary cultures. And with no surprise, we found more splicing defects in the astrocytes in comparison to neurons, because our primary neurons, at least in culture, they express lower levels of DMPK and they accumulate less RNA foci. The splicing defects were mainly concentrated around events that involve exon skipping. And when we did a gene ontology analysis, we found that they were enriched for transcripts associated with the organization, the dynamics of the cytoskeleton, that will therefore have an impact on the cell morphology. That also comes up as a GO category in our analysis. And both actin and tubulin cytoskeleton seem to be affected. So we, by RT-PCR, standard RT-PCR, we confirmed some of these splicing defects in the primary astrocytes that could be recreated in astrocytes knocked down for MBNL1 and MBNL2, again supporting the role of these splicing regulators in the spliciopathy seen in the astrocytes of our mouse model. But we were worried that this was an artifact of the cell culture and somehow we were inducing stress or some sort of artifact that was not representative of the in vivo situation. So we went back to the mouse model and we isolated cortical astrocytes from adult mice or young adults between 1 and 2 months of age. And you can see here, that the splicing defects in the astrocyte fraction, and I will focus on integrin alpha 6 here, which is one of the transcripts that shows the most severe splicing defects in our mouse model. You can see that there is a clear exclusion of the exon 27, I think. Yeah, 27. There's a clear exclusion in the astrocyte fraction that is not very obvious in the cell flow through that consists of other cell types that are not astrocytes. And of course, when we do the same analysis in a whole tissue sample in the frontal cortex of the same animals, these splicing defects become less pronounced because now we are dealing with a mix of different cells. Some of them are most likely less affected than the astrocytes. So the defect of the integrin alpha 6 now here is less obvious than it is in the specific astrocytic fraction isolated from this tissue. So it is known from the literature that during the maturation and the differentiation of different cells, including the astrocytes, multiple transcripts, hundreds of transcripts undergo a physiological splicing transition. So we crossed our data, we crossed the splicing defects that we see in the DM superXL astrocytes with the transcripts that undergo this natural transition under physiological conditions. And we found a significant overlapping. And more importantly, when we looked more carefully at the type of splicing defects, we found that most of them, the majority of them, reflected the increased expression of isoforms that are typical of immature cell types. Let's focus on the exon 27 of SORBS1 here on the top. You see that during the maturation of the wild type astrocytes, so under normal conditions, these exon, the inclusion of this exon increases over time. although this inclusion does not take place in the DM superXL astrocytes. So they retain the expression of an isoform that is typical of the immature cells. And this is true to a greater or less extent to all the other transcripts that we have included in this analysis. So the DM superXL astrocytes, they keep this molecular signature of impaired differentiation, suggesting that their terminal differentiation is somehow blocked due to the expression of these toxic RNAs and the dysregulation of the MBNL proteins. So we then moved into human samples to see to what extent were we looking at something that is relevant for the disease. And of course, we confirmed the accumulation of RNA foci in the astrocytes that seemed to be even a little bit more abundant than in neurons, with the downregulation of the GFAP. And in humans, what is also interesting is that there is not only a downregulation, but there seems to be a difference in the isoforms of the protein that are expressed in DM1 frontal cortex. We still don't know which isoforms they represent and what this means in physiological terms. And we confirmed the dysregulation of the splicing of the same transcripts that were altered in the DM superXL brains. And when working with a glial cell line, that was our best model to mimic astrocytes in culture, we found that upon expression of the CTG repeat and accumulation of the RNA foci, the induced cells could also reproduce this adhesion phenotype and this delayed spreading in culture. Okay, good. So we have astrocyte effects, but how does this affect the neurons? We don't know, and I will not give you the answer today. I would just like to remind you to bring back something that we found in the past. So we found in the past, again, in the DM superXL animals, a pronounced accumulation of RNA foci in a confined population of astrocytes in the cerebellum that was easy to detect just on histological sections. That is the Bergman glia here that surrounds the Purkinje cell of the cerebellum. And this was associated with an abnormal activity of the neuron and some defects in the motor coordination of the mice. So this was somehow evidence that there might be non-cell autonomous mechanisms operating in the brain of the disease. So defects in astrocytes will impact the biology of the neighboring cells. And in culture, we can do funny things. We can mix different cell types together with different genotypes and see what is the outcome. So here what we did was to grow neurons on top of astrocytes of different genotypes. And we found that regardless of the genotype of the neuron, whenever they were growing on top of a DM superXL astrocyte, they always showed delayed maturation of their neurites, their projections. So the presence of DM superXL astrocytes impacts somehow the development of the neurons. But as I told you before, we also know that the DMPK gene is expressed in other types of in other cell types of the brain, including the oligodendrocytes or the oligodendroglia, because it can go from OPCs, the progenitors, to the mature myelinating, fully differentiated oligodendroglia here on the right. So the DMPK, the endogenous DMPK, is expressed in this cell type. So we asked, does this affect the myelin of the DM superXL animals or the myelin in DM1 in general? So we first started this study by focusing on a region of the mouse brain that is known to be very rich in myelin, the corpus callosum, that is here in purple. That is like a bundle of myelinated fibers that connects both hemispheres and allows the communication between both halves of the brain, where we could confirm the presence of RNA foci in oligodendroglia here that co-localize with MBNL1 and MNL2. But what are the physiological consequences of these? So the first thing we noticed that was already cool was that the thickness of the myelin in the DM superXL mice was reduced, but only at 2 weeks of age. You can see here that the corpus callosum, labeled here by the myelin basic protein, is thinner. Well, it's actually quantified here, it's easier. It's thinner than in the wild type controls. And this is most likely due to a significant reduction in the percentage of myelinated axons at this early time point in comparison to the wild type controls. However, the phenotype recovers itself at later stages. We do not see this difference at 4 months of age, suggesting that there is a delayed myelination rather than a total block in the myelination of these mice. And this goes together with the peak of expression of our transgene in the isolated oligodendroglia from the corpus callosum that seems to be highest at 15 days, or 2 weeks, the time point when we see this reduced thickness of the myelin. Then the transgene expression goes down in the oligodendroglia from 15 days onwards. This delayed myelination is confirmed by the analysis of myelin proteins like MBP and CNP, which are significantly reduced at 2 weeks and 1 month. And then the difference is no longer detectable after 4 months of age. So again, confirming the transient defect in myelination in our mouse model. But how do we explain it? Why at 2 weeks we do not have enough myelin? So to answer that question, we have taken advantage of markers of different differentiation stages of oligodendroglia. And we found that there was an overall decrease in the oligodendroglia at 2 weeks of age, here labeled with OLIG2. You see that there is a decrease in the overall population of oligodendroglia in the corpus callosum. But more importantly, there was an increase in the progenitor cells, so the oh no, what have I done? Yeah, in the OPCs here, there is an increase in the OPCs here labeled by PDGF receptor alpha in the DM superXL animals, and a decrease in more mature stages of the oligodendroglia lineage, here labeled by CC1. So it looks like there is a blockage or a delayed differentiation that leads to an accumulation of OPCs at this early time point, at 2 weeks of age of these mice. And at this cell stage, the OPCs are still proliferative, and they are before committing themselves into the full differentiation. And we have confirmed that the cells that are accumulating here are actually proliferating by co-labeling with Ki67. And you see that the OLIG2 co-labeling with Ki67 leads to an increase in the DM superXL mice, again, supporting this arrest or this delayed accumulation of cells in this precursor cell stage. So we turned into cell cultures again to get a better insight about these. And during differentiation of OPCs in cell culture, you have these dramatic changes and ramification of the cell projections from a bipolar cell morphology to an extremely ramified oligodendrocyte. And by live cell video microscopy, again, we showed that the OPCs from the DM superXL animals, they developed fewer branches and shorter branches over time, again, in support of a impaired differentiation. That goes together with an accumulation of foci that now is slightly different from the in vivo situation. The DMPK transgene increases over time, and the foci increase over time. And we can discuss why this difference with in vivo later. So we used RNA sequencing to find the targets again. And of course, there are many more splicing disregulations in the oligodendrocytes in comparison to the OPCs. And like in astrocytes, the transcripts are primarily affected play a role in the morphology and the cytoskeleton of the cell. And the splicing defects that we see also increase the expression of isoforms that are typically of immature cell stages, again, suggesting that all these leads to an impaired terminal differentiation of the cells. They are committed to become oligodendrocytes. They get there eventually, but they have problems to get there. So, and finally, we wanted to have some human data on the oligodendrocytes. So we collaborated with Cécile Martinat to get iPS cells and to differentiate them in oligodendroglia to be on the safe side, oligodendroglia that accumulate RNA foci, and that when we label their ramifications, we see that they are less ramified than the control oligodendroglia from healthy donors. So to conclude, the RNA toxicity leads to some sort of hypertrophy of the astrocytes that is critical because these projections that the astrocytes form and that go around, wrap the synapse are very important to maintain the homeostasis of the synapse. So if the astrocytes are shorter, are hypertrophic in DM1, the control of this environment will be less efficient, and so the neurotransmission will possibly be less efficient as well. In the oligodendrocytes, we have also morphological defects that will translate into a delayed myelination, that it's transient, and even if it recovers, because it happens during a critical postnatal window that is so important for the establishment of neuronal networks, even if it recovers, it may leave consequences to the function of the brain at later stages. And in both cases, these changes are associated with RNA missplicing that affects mainly transcripts of the cytoskeleton, of cell adhesion, and that are involved in the morphology of the brain. And that they seem to be dependent on MBNL function, on the regulation of MBNL, and that leads to splicing profiles of immature cells. So we are now interested in understanding better what are the subtypes of astrocytes and the subtypes of oligodendrocytes that are affected, where in the brain they are affected, and what are the direct role of these cells in the pathology of DM1. And for that, we are taking advantage of a new conditional mouse model that we have developed in the lab. But regardless of the answer to those questions, one thing I think we can say already is that we cannot forget that in the future therapies targeting the CNS, we should not overlook the non-neuronal cells. They should tackle not only the neuronal defects, but also defects happening in other cell types of the brain to be fully effective. And with that, I would like to thank to the people involved in this work and my collaborators, and as well as the people that paid for the bills and that allowed me to be here today. And I thank you, and thank you for your, and I'll take questions.
Yeah, we have time for a few questions, and I'm sure Mario will be available later in the day to talk to. Yeah.
Really great talk. So these splicing events that you're observing are nonetheless a correlation with these morphologic impairments. And just as a potential contributing mechanism, it's well established that RNA localization and local translation play roles in both of these, or the oligodendrocyte, early myelination, as well as these, astrocyte enphy. And I was just wondering, have you ever done immunofluorescence to see if MBNL is in these peripheral compartments?
No, we haven't. This is something we are very interested in particular for the MBP. MBP, MBP, for instance, is local translated. So a change in the localization of, because the downregulation of MBP, we could not correlate with the downregulation of the overall transcript. So it might be just that is not well localized within the cell. So you're perfectly right. There's something we need to look into. Yeah. And other transcripts as well. Many of the cytoskeleton, even I think actin is locally translated in some parts of the cell. So yeah.
Take MBNL or GFP and just see if it goes out there. I wonder if it is.
Yeah.
Oh, there's a question back here. Maybe that one. Thanks.
Great talk. I was wondering if you were able to look into microglia, like the role of microglia in these oligodendrocytes.
Yeah, well, the short answer is no. We didn't have a look yet. We did at some point did a quick immunostaining to see if microglia had foci. I would not swear that this is the right result. We didn't see many foci in microglia in our cells. We didn't see an over-proliferation of microglia in our mouse model, but this is one single shot. I don't think I can comment on that. We should do it again properly. This was just a one-shot experiment.
Mario, that was great. Thank you very much. I don't know enough about glial biology. Is there any ongoing turnover of the glial cells? And what I'm interested in any somatic mosaicism between glia and neurons, and whether or not you're seeing something similar in humans and in mice in that regard.
Well, these are two good questions. For each one, I don't know the answers. Or what I know is that microglia, glia, they do proliferate in the adult brain, but not much, I think. Astrocytes can proliferate, but they're not. It's rare, and of course, then oligodendrocytes can proliferate, but following an injury. There's always this pool of OPCs in the brain, but they need to be reactivated, and there are even strategies in MS to try to reactivate those to myelinate the brain again. But I'm not very knowledgeable on the topic. For the second question, it's a very good question. We don't know yet, at least in myotonic dystrophy, the differences in the somatic mosaicism between different cell types. This has been done in Huntington's. In DM, I don't think it has been done now in brain cell types. No. But we do not. But to connect the first question with the second question, we do not need cell division to have somatic mosaicism. So it doesn't. In Huntington's, it was shown that the neurons have higher expansions than the non-proliferating cells in the brain. Than the proliferating cells in the brain.
That's right. Yeah. Vision of it's actually selected against.
Yeah. Okay. Yeah.
Thank Mario one more time.